All right, looks like it's probably time to get started. So I'll go ahead and give an introduction. So it's my privilege to introduce today's seminar speaker, Dr. Xinxin Ding. To start by giving you a little bit of background, Xinxin received his Ph.D. in biological chemistry from the University of Michigan in 1988. His dissertation advisor was Dr. Judd Kuhn, who was certainly one of the giants of early giants in the cytochrome P450 field. After receiving his PhD, Xinxin stayed at University of Michigan for a few years as a postdoc, lecturer, research investigator, and finally as an assistant professor. And then in 1994, he moved to Albany, New York, where he took a position as a research scientist at the Wadsworth Center at the New York Department of Health, as well as a faculty position at the State University of New York at Albany, so SUNY Albany. There he rose through the ranks, and it culminated with his being a professor and director of the Laboratory of Molecular Toxicology in the colleges of nanoscale science and engineering at SUNY. In 2017, Xinxin accepted the position of professor and head of the Department of Pharmacology and Toxicology at the College of Pharmacy at the University of Arizona. And he holds a position of the R. Ken and Donna Coit Professorship in Drug Discovery. He's a member of several centers at the University of Arizona. Quite notably, he's director of the University of Arizona Superfund Research Program P42 Center. And he's also a member of the Bio5 Institute, the University of Arizona Cancer Center, as well as a member of the Southwest Environmental Health Sciences P30 Center, where he actually does the same job I do in CURES. He's a leader of their pilot project program. Xinxin's had continuous funding for his research from NIH throughout his career. He's author of more than 200 peer-reviewed papers. He's a couple other points of note. He's editor-in-chief of the journal Drug Metabolism and Disposition, and he's currently the secretary-treasurer of the American Society for Pharmacology and Experimental Therapeutics, so ASPET. As far as his research, broadly described, his research interests are in drug metabolism, chemical carcinogenesis, and mechanistic toxicology, and their application in drug development, disease prevention, and precision medicine. And I would say specifically, he's been interested in the role of cytochrome P450 in xenobiotics toxicity in various extrahepatic tissues, including tissues of the respiratory tract. And that's really where his seminar is going to be focused. So I'll turn this over to you, Xinxin. You can see the title of his talk right there, Metabolic Mechanisms of Chemical Toxicity in the Lung.
Thanks, Tom. Thanks for the opportunity to connect again. It's been a long time since I visited Wayne State, but I've known Tom, Melissa, and Haisuk and other people for a long time, and I wish I was there in person to get to talk to people more. But this is great to at least see some of you and to give you an update on what we've been doing. So, this is always okay, good. And as Tom said, I am a P450 guy, so I work on P450. For those who are not familiar with the P450 enzymes, so these are this represented by this red block, and they need a P450 reductase as abbreviated as cyclone CPR or POR, stand for P450 oxidoreductase and cyclone P450 reductase, respectively. So there's lots of P450s, but only one reductase. So my lab has worked on a number of P450s, but mostly we worked on these. So this slide was from Fred Gingrich of Vanderbilt, and sort of sorted the different human P450 enzymes into based on the main substrates they metabolize. So most of my work really have to do with the CYP2As, 2A6, 2A13, 2F1, 2B6, and maybe a little bit. And then recently we've sort of done a little bit interesting stuff on the CYP19, which is the aromatase that's involved in estrogen synthesis. So this is a sort of my pitch slide when I talk to students who try to recruit people, but kind of summarizes what we do. So the P450 enzymes obviously have a lot of, because the many different forms and different genes that metabolize a lot of different substrates, including external compounds like drugs and carcinogens, also endogenous compounds like I mentioned, estrogen, steroids, and so on. So our approach has been to develop knockout mouse models and transgenic mouse models to manipulate the P450 enzymes, be it knockout from the mouse or put the human ones in there, and so on. And to look at both look at the role of P450s in drug toxicity, drug metabolism, or carcinogenesis, and so on. So as part of this, we've developed capabilities in inhalation exposure. So I actually run the resources for the P30 Center here for inhalation exposure, where we do variety of exposures like tobacco smoke exposure, dust exposure, and we're trying to develop a wood smoke on account of recent surge in fire smoke exposures and concerns by the fighter safeties and so on. So we've also been trying to identify the function of P450 mediated metabolism of endogenous compounds, and mostly by knocking out the P450 reductase, which if you knock out the reductase and the P450, none of the microsomal P450s would be functional. So we've looked at a bunch of different tissue-specific knockouts like the brain, heart, and so on. So this this little slides here show the latest thing that's on the kidney. We found a phenotype in the kidney where mice with low P450 reductase expression have vacuoles and impact the kidney function. And so we've been doing a little bit with that. But in terms of toxicity, though, most of the chemical toxicity, we've been focusing on the lung, looking at airway epithelial cell injuries and repair. And over the years, then we've also developed capabilities in metabolite analysis, looking at using mass spec to look at different metabolites, protein adducts, DNA adducts, and so on. So I'll talk more about this aspect of that. And so I guess today's talk, though, I will focus on the metabolic activation, the metabolic mechanism, namely the formation of reactive metabolites and what controls the fate of these metabolites, because that's something we've been really focusing on. So you've probably seen this scheme in a textbook where biotransformation of foreign chemicals, environmental chemicals or drugs undergo phase 1 and phase 2 components, and phase 1 mostly is P450 enzymes. And I'm sure you've heard about phase 2 as well because of what Tom and Melissa's working on over there. But we all know that many of the toxicities induced by these chemicals require bioactivation to form reactive metabolites. So there's not really much new there, although these reactive metabolites, because of the chemical nature, their reactivity, it's hard to study because they're unstable. You can't really isolate them, and they're with many of these compounds. But another thing is that most of the studies so far has considered really focused on the ability of the reactive metabolites to cause toxicity within the cell. Much less is known about the ability of these metabolites to move out of the cell where they're produced to neighboring cells or get into the circulation and reach remote organs where they might cause toxicity. And we kind of got drawn into this, and because of necessity, as we study, we do risk assessment for chemical toxicity in the lung, in considering the relative contributions of the lung versus liver. As we all know, the liver has a lot more P450 enzymes, other enzymes that produce a lot of more reactive metabolites. So that's the sort of the framework of what we've been working on, and I want to talk about. Now, so we've looked at a number of different chemicals that can cause lung toxicity. And I started with acetaminophen, which most people know as a hepatotoxicant, but it really does also cause toxicity in other or in extrahepatic tissues like kidney, lung, and so on. And I've also done a little work with paraquat, which induces lung fibrosis, in addition to Parkinson's disease. NNK, which is a tobacco-specific nitrosamine, so it's a tobacco-associated carcinogen, and a bunch of other nitrosamines. They all undergo P450 mediated bioactivation. And monocroptolene is a alkaloid that's been found in plants that are known to cause liver toxicity as well as extrahepatic toxicities. A lot of our work is focused on naphthalene as a environmental pollutant, and I'll focus on this compound a lot. And we've done a little bit with tobacco smoke exposure-induced lung cancer, and then lately, as part of the Superfund program, we've been studying arsenic toxicity. So let's start with acetaminophen. And this, again, is textbook stuff that you probably know, but let's look at this just as a review. So acetaminophen is metabolized by P450 to form this reactive metabolite, NAPQI, which can interchange form the N-hydroxy, a little bit more stable N-hydroxy compound. But this is detoxified by conjugation with glutathione to give you this conjugate. And the liver cells and most cells have plenty of glutathione, so usually this is not a problem. But if you have overproduction of this compound, this reactive metabolite, because of overdose or because of increased bioactivation, and this could escape glutathione and start to react with protein, and that's the mechanism that leads to cell deaths and patient would die. So we asked this question because when I started on looking at the P450 function, try to develop the knockout mouse models and so on, we use APAP as a model toxicant. And so since I was focusing on extrahepatic tissues like the kidney, lung, the nasal mucosa, which some people don't know that actually has lots of P450 in it. So we asked the question, okay, the toxicity induced by acetaminophen, and you can see in this wild type versus acetaminophen treated mice, you can see the toxicity induced in the kidney, in the lung airways compared to the vehicles, and in the olfactory mucosa. Did this come from local bioactivation, or did this come from metabolites that's formed from the liver? And so we made this mouse model that had the P450 reductase conditionally knocked out. So this is the reductase, the CPR gene. We put the 2-LOX-P site into the introns, and by crossing this to a albumin-free mouse, we were able to specifically knock out the reductase from all hepatocytes in the liver. So this is a liver CPR mouse model. And we found that, yes, in this knockout mouse model compared to the wild type, this LCN is this liver CPR knockout mouse model, you no longer see APAP toxicity. So clearly the kidney toxicity requires bioactivation of APAP in the liver. And in the lung, I mean, there's some toxicity still, but they're not as bad as in the wild type. In the nose, though, there's really not much quantitative difference. So this says, okay, clearly the liver-generated metabolite can contribute to toxicity in at least some organs, particularly in organs where it didn't have as much metabolic activity of its own. So that was interesting, although we didn't really know what the metabolite is, because we're looking at toxicity, which doesn't tell you which metabolite caused that toxicity. Now, we did we had an opposite finding with NNK, which is a well-known lung carcinogen. And it's a nice bioassay in mice that was developed that can assay for NNK-induced lung cancer. So this scheme mostly developed by Steve Hack's group and the others colleagues currently at University of Minnesota depicts the bioactivation of NNK. So NNK is metabolized by P450 at either of these alpha carbons. So you get two different pathways coming down, but without going into too much detail here, but both pathways would either attacking this carbon or this carbon would give you this reactive diazonium ion that can cause DNA adduct formation. And formation of DNA adduct is important for NK-induced lung cancer. So we made a knockout mouse model where we knocked out a bunch of P450s in the CYP2 gene cluster, including the CYP2F2, 2A5, and all the CYP2Bs, and all the CYP2As, and so on. Now, these are the enzymes that are already known to bioactivate NNK. And we show that in the knockout mouse, you can, wild type, you can induce tumors, lots of lung tumors with different dose in the lung, but in the knockout, you can no longer induce lung tumors. So that means that these P450s are important. Now, of course, then what's the organ contribution? Did this tumor genesis come from bioactivation in the lung or not? And I won't go into the details because these are all well-published stuff. But basically, we looked at a mouse model that has reductase knocked out from the lung versus one that's knocked out in the liver. And yeah, the one knocked out from the lung affected the lung tumor genesis, but the one knocked out from the liver actually increased lung tumor genesis because the pharmacotoxicokinetic interactions. But so liver bioactivation of NNK did not play a major role, at least in lung carcinogenesis. All right, so moving forward then, recently we've also done some work with paraquat-induced lung fibrosis, because this is supposed to be a paraquat is metabolized by a number of reductases, and we thought we want to check out P450 reductase, see if that had any impact. And so the answer is yes, the bioactivation in the lung is actually important for paraquat-induced lung fibrosis. And we've also looked at the this alkaloid-induced lung injury. So this is actually a class of compound that has been found in populations that accidentally ingest plants that have these high levels of this alkaloids, and they cause deaths through liver injury and also cause lung vascular damages. And so we found that with the lung and liver reductase knockout mouse models, well, actually with the liver reductase knockout mouse models, we found that this bioactivation in the liver is important for the toxicity, the vascular toxicity in the lung. So all of these are fine. I mean, it's a very nice way to at least to sort out if the remote contributions is happening or not, but it doesn't tell us what the metabolites are, even though you can sort of predict. And so now to focus on naphthalene, where we actually had a handle on the kind of metabolites that are formed. So why naphthalene? Naphthalene is a it's a mothball, main ingredient in mothballs. Everybody have some exposure probably in the past, but it's also a major pollutant. It's in airways, it's in gasoline products, it's in tobacco smoke. And recently, it's found that firefighters actually have naphthalene metabolized as the highest contaminant in their blood and urine. So it's pretty high ubiquitous exposure in the population, and it's a very nasty chemical in the sense that you can, at the occupational exposure limit, the OSHA occupational limit, which is 10 parts per million, you can easily induce lung injuries, lung airway, epithelial cells sloughing off, the cleft cells get squamated and vacuolated in mice with just one setting of a couple hours of exposure. So that's a real pretty bad thing. And then the EPA has been trying to do risk assessment for those for repeatedly. And it has been classified as a possible human lung carcinogen because the National Toxicology Program did 2-year bioassays and found it causes a lung tumor in mice and lung and nasal tumor in rats. So there's a lot of interest in it as well, in addition to being a model sort of bioactivate toxicant. And recently, an unpublished data from NIEHS is showing that chronic exposure to occupational exposure to naphthalene also induces lung fibrosis. So this metabolic scheme, we'll go through this, but this most of this work was done by Alan Buckpitt's group, who Alan is retired some years ago now. They really mapped this whole metabolic scheme out very nicely. So here's the naphthalene, P450, mostly it's CYP2F, 2A enzymes, although a bunch of other P450 also has activity in converting this to a epoxide. And so epoxide can be detoxified by conjugation with glutathione. So that's a major, one of the major ways of detoxifying this. But epoxyhydrolase supposedly hydrolyzes, and then this will further get converted into the dihydroxy and then the quinol, the 1,2-naphthylquinones. So the color scheme here is that the red compound usually means it's a reactive compound. It's I mean, obviously differ in the reactivity. Some are more reactive than others. So epoxide is chemically probably most reactive of the bunch. So I don't think we can measure this directly. But then, so these quinols actually you can measure. You can it's relatively stable, although I mean everybody probably knows that they're reactive. So you can also coming down this pathway to form first the naphthals, which is one of the major metabolites you can detect in human urines or their conjugates to through phase 2 reactions. But this can be further metabolized to give you 1,2-naphthylquinones. And this diol, the hydrodiol, can be further metabolized up by P450 to give you the diol epoxide. So there's at least 1, 2, 3, 4 different reactive metabolites could potentially cause toxicity. It's not really known which one is important as far as lung toxicity is concerned. So one of the major controversies from this in the risk assessment is that the CYP2F enzyme, mice has the most reactive one. I mean, it's Tom knows about this, that this P450, the efficiency of catalysis for CYP2F4, 2F2, which is the mouse isoform, mediated naphthalene metabolism is like huge. It's very fast. Whereas the human form, which is CYP2F1, just originally cloned by Frank Gonzalez group and then shown to have some activity, but nowhere near the kind of activity you see with the mouse CYP2F activity. So based on that, the chemical industry has been arguing that the toxicities that you see in mouse, particularly for naphthalene and styrene, which is another compound that's related to structurally related to this and then has caused similar kind of lung toxicities, is not relevant to humans because humans enzymes are not that active. And so there's need to look into details. So we did this work a few years back and it was published. And we made this human mouse model where we first would knock out the CYP2 cluster, as I've shown in the previous slide. And we put the human CYP2A13 and 2F1. So these are the orthologous forms of the mouse CYP2As and 2Fs into this mouse so that they produce the human P450, not the mouse P450. And we tested these mouse compared to the wild type for naphthalene-induced inhalation toxicity in the airways. So in the nose and the lung, the nose is actually more the toxicity is more robust here. Wild type exposed to 10 ppm naphthalene, you can clearly see the olfactory region of epithelium is all sloughed off or it's damaged, undergoing necrosis. But if you look at the knockout mouse model, this is all intact, nice pseudo-stratified olfactory neuronal cell layers and so on. And in the humanized mouse, where you had the human, which would be equivalent to the wild type because they have the human P450 of equivalent of the mouse, yes, you still see these toxicities. So this shows that clearly the human 2A13 and 2F1 are capable of doing the same damages to the airways that's mediated by the mouse P450. And we also in the lung, that's the toxicity is less subtle, partly probably because of the density of P450s in the mouse lung, and because the exposure dosimetry, there's less at the lower levels are encountered here. That you do see toxicity and compared to the knockout versus the humanized, and there's the cells are more change. You see vacuoles, all that. And if you count up the sort of the volume fraction, which is a way to count the number of damaged cells in the airway, basically you count out all the damaged cells over the total cells. And this is a filter air control. This is 10 ppm naphthalene. You can see the humanized produce a lot more damage compared to the knockout. So again, this shows that these human P450s are able to cause harm, mediate naphthalene-induced toxicity. Now, another thought related to this risk assessment is that the point whether naphthalene metabolites in the liver, which produce first the naphthalene oxide and then other metabolites, as I showed in the scheme that has the 4 different reactive metabolites, how many of these can actually come out to the blood and come to the airways and the lung to cause toxicity, right? I mean, you have inhalation exposure, naphthalene can directly contact epithelial cells, but very quickly we know that the naphthalene gets to the blood, gets to the liver. So really, there's not a barrier for naphthalene to get to the liver to undergo bioactivation there. So we first look at the cytotoxicity in the lung compared to the wild type in the knockout mouse models we have. So this is a little busy here. So this is looking at a lung reductase knockout versus wild type. And we're using so inhalation exposure mice to 10 ppm naphthalene, and we wait 2 days for the toxicity to develop, and then we label it with BRDU to label the proliferating cells. So this is in different parts of the lung. This is the proximal region, larger airways, and this is the distal, smaller airways, to look for the BRDU staining, which is a marker for proliferating cells and proliferating because they were injured. So this is just to remind people, this is the reductase knockout where the bioactivation in the lung, most of lung is turned off. And you can see, and this is the controls, this is in naphthalene exposed mice, that in this blue, well, it's a blue, I guess I'll call it blue bar, at the reductase knockout, there's significant reduction in BRDU labeling, which means that there's less cells damaged or proliferating. But it didn't go all the way down. So there's still significant amounts in there. So at least part of the remaining ones could potentially be contributed by circulating metabolites, right? So we look at the opposite model where we knocked out the P450 reductase from the liver. And now, so ignore this part because you've seen that. Here we're looking at volume fraction again to see the damaged cells, the fraction of damaged cells over total epithelial cells. Control 5 ppm naphthalene, 10 ppm naphthalene. So the blue bars are wild type and the green bars are knockout. And you can see that the knockout has a significant reduction. I mean, not totally reduced, definitely, but it's a significant reduction compared to the wild type in the cells damaged. So you turn off metabolism in the liver and you see a decrease in toxicity in the lung. That seems to me to be saying that the liver is contributing some of the metabolites to the lung to cause toxicity. Now, as a control, we look at the pharmacokinetics of naphthalene itself, in the knockout, actually you see an increase in naphthalene levels because the clearance, metabolic clearance is slower, which is what we would expect. So this decrease in toxicity is not because we didn't have enough naphthalene going in. So what this means is that the question, does hepatic bioactivation contribute to cytotoxicity in the lung, is the answer to that is yes. Well, we don't know which one of these four metabolites are actually contributing to this, but liver is contributing. Now, what the significance of this finding, though, is that suddenly risk assessment no longer it can be relying on just the lung, relative metabolic activities in mouse and humans in the lung, because you need to consider human versus mouse liver metabolism of naphthalene and the ability of these metabolites that can go from liver to the lung. The human liver is just as active, if not more active than mice in terms of bioactivation of naphthalene and other compounds, and as we all know. So that kind of is something I think it's important to consider. All right, so now lately one of my students decided to look at DNA adducts of naphthalene. So naphthalene has been known to be primarily a non-genotoxic carcinogen, in that the mechanism carcinogenesis is because of this repeated injury and repair. You one exposure within 24 hours injured, and then if you wait a couple weeks, it's repaired. So the cycle of injury repair over chronic exposure would promote non-genotoxic carcinogenesis. But well, genotoxicity also occurs. So the question of whether genotoxicity is important for carcinogenesis is something we're actively studying. So Sarah Hannon is the student who's working on this, and then she has detected a number of DNA adducts in mice in different tissues in blood, lung, and liver, and so on. We identified 7 different adducts here. I won't go into details, but these are mass spec showing the detection of these adducts at different sort of mass transitions at the same retention time, and then we sort of validate this by synthesizing standards and so on. The bottom panel here is showing a dose response, and mice will be exposed to 0 to 200 milligrams per kg injection of naphthalene, which is a more sort of controllable exposure mode. And you can see this DNA adducts for this. I think this is for the 4 different stable DNA adducts, increased proportion to dose in blood, white blood cells, in lung tissue, and in the liver. So clearly it's formed in vivo in the target tissue, and the P450 dependence of this thing, and which kind of helps with validating, and these are real adducts and not just some artifacts you detect, because they're P450 dependent. So when we compare wild type to the CYP2 ABFGS knockout mouse model, look at this DNA adducts. This is for one of the DNA adducts as an example showing here, but it's similar for all adducts we've looked at. And you can either wild type or knockout. This is the control. There's no adduct in the control, this first bar here. And wild type, if you treat with 200 or 300, you get the blue and orange bar, I guess. You get plenty of adducts in 3 different tissues. And the knockout, which you can't see here at the last 2 bars, you don't see adducts, nothing much anyway. So this again is confirming that these adducts are real, are formed by P450 mediated by activation. We also detected, so those adducts were naphthylquinone formed adducts, and these are naphthalene oxide or epoxide formed adducts. We also detect these in there, and I won't go into details. So I know you're waiting to see, okay, what's the obvious question is, okay, is liver metabolism important for DNA adduct formation in the lung. Now, this time we would be able to tell which one is the metabolite is actually involved, because the DNA adduct tell you what metabolite is bound to the DNA. Well, we're not there yet. We're waiting for the mice to do this crucial experiment to compare wild type to liver CPL knockout mouse model. And so this will tell us, though, whether this liver is contributing to either NAO or NQ metabolites or both or no. And we're sort of holding our breath for that. But meanwhile, we did do another experiment sort of support this idea that the liver generates an Na oxide, maybe transport to the lung to cause toxicity. And this may be helped with non-covalent binding to serum albumin. So again, this scheme here, the metabolite, let's say focus on naphthalene oxide formed in the liver. And it's a reactive compound. It's like if you just look at it in the test tube, the half-life is probably a minute or two. So it's very unstable. It will go to the lung, and where they can cause react with DNA from DNA adducts. And so why do we think albumin is involved? So we made this, long story short, we made this albumin humanized mouse model for another project to sort of develop biomarkers of albumin protein adducts with different chemicals and tobacco smoke and other compounds and with drugs but in the process, and we had to sort of use this albumin knockout mouse model as a background, and that to put the human, because the human and mouse albumin actually have about more than, I think, 30% difference in amino acids. And certainly there's one critical peptide as a cysteine peptide that's not present in mice, but it's in humans. But anyway, so we compared the wild type and the serum albumin knockout mouse model. So the key here is you got 4 groups: wild type filter air, wild type naphthalene, albumin knockout, filter error, albumin knockout, naphthalene. And then we look at this the 3 DNA adducts formed, stable adducts formed with naphthalene oxide. So the blue bar here is the naphthalene. So you see adducts in here, and then this orange bar is the knockout. So the wild type knockout, wild type knockout. And you can see across the board, there's a trend, at least a trend of decrease. And in some cases, in half of these cases, a significant decrease in the DNA adduct detected in liver or lung and in the knockout mice. So this is suggesting that serum albumin is somehow binding to these reactive metabolite, stabilizing and helping it to get to, or allowing it to get to the lung. And when you don't have the albumin to stabilize these, obviously you have some other proteins that can probably do the same thing, but albumin is the most abundant serum protein in mice and humans. So, so this proves that maybe the reactive metabolite formed in the liver does get to the lung. All right. All right, to summarize, study the mechanism toxicity can be complicated because the bioactivation can form a variety of different reactive metabolites, and they're not stable, and that's not easy to study them in a sort of in the test tube. The use of transgenic knockout mouse models can help, at least give you answer some of the questions in steps, although it won't solve all the problems for you. And what we found, though, with the different toxicants we've studied, that the mechanism of toxicity can vary for different chemicals, different toxicants. It depends on the relative stability of the reactive metabolite, the enzymes involved, and all that. And some even for different toxic endpoint, like cytotoxicity versus genotoxicity, that could be different. And I just want to reiterate that the knowledge on organ and enzyme contributions are important for risk assessment and prevention efforts. For instance, In the case of NNK-induced lung cancer, I mean, since it's looks like exclusively dependent on bioactivation in the lung, so if you try to prevent tobacco smoke-induced lung cancer by inhibiting a P450, you should probably try to do a inhalation exposure directly target the lung rather than the liver. Whereas in cases where the liver is making a major contribution, you may consider trying to inhibit systemically or target the liver. So again, before I stop, I want to just to say that we have a number of mouse models made, different P450 knockouts, reductase knockouts. We also have epoxy hydrolase, microsomal epoxy hydrolase conditional knockouts now. We have the albumin humanized. If anyone wants to use these mice, feel free to contact us, and we've been giving them out. So just a little promotion here for that. All right, I want to acknowledge people who did the work. So my group, we had a number of people who worked on this NAFLING, and now the other stuff we've published. So I won't go into the details, but Sarah Hannon is the one did most of the DNA ad hoc work. Natalia Kovace, who now works in industry now, has done a lot of work with the NAFLING pharmacokinetics and cytotoxicity and so on. I also heavily collaborate with my wife's group. Chin Yuzhan is my wife, who's also a faculty here, and people in her group contributed to these projects also. And Laura Van Winkle from UC Davis is my longtime collaborator. She brings the expertise in inhalation toxicology, cell biology, and so on, and has been sort of part of the legacy NAFLING study group for a long time. And also Bruce Buckholz from Lawrence Livermore, who's been helping us with looking at DNA adducts using C14 label NAFLING. And Jeff Burgess here we're starting to look at NAFLING adducts in human firefighters. In fact, we've detected the NAFLING adducts in human firefighter urines and blood samples. And Rob Tureski at Minnesota was my former colleague at Wadsworth in New York, and who helped us and really taught us how to use mass spec and has been very instrumental. And our funding mostly comes from NIEHS and from NCI, and the P30 Center, the Cancer Center here, and the Superfund Center all contributed to the work. And there are a few slides here showing the Tucson, Arizona. This is a view from my roof that you can see, and when the rain comes, the mountains do turn green, although most of the year it's kind of grayish looking. As our new addition to our building here. So welcome everyone to visit. I'll be happy to take any questions, I guess. Thank you for listening.
Thank you, Xinxin. Are there any questions? Graham, I see your hand up.
Yeah, thanks very much. That's a fascinating presentation. I wondered, I remember many years ago now seeing a great presentation by some researchers at Wake Forest that were trying to put together, as far as I could work out, a body on a chip by having various organ tissues. And that what they were demonstrating very elegantly was that if you take a certain toxicant and you threw it at cardiac tissue, there was no problem. But if it had been through lung tissue first, then you saw the problem that we saw, you know, in the real world with real people. I wondered, have you explored any such preparations yourself?
Thanks. That's a great question. I mean, this is the sort of the alternate and maybe even better model than the mouse model that we're using, because certainly it's going to be a lot quicker and cheaper once the chip is made. And there have been a lot of, in fact, at the SOT meeting this year, I mean, a lot of booths there that's selling chips, organ on the chips. I mean, it's very simplistic, and many of those just some different cell types on the cover slip and so on. But there's some really good ones. We haven't really done any work with the chip, but certainly this approach in theory, could work. I mean, if you have the livers, the hepatocyte, if you can culture them in a sort of 3D format that can recapitulate the metabolic capacity of the real liver, and you can have the lung cells can represent what the lung look like in vivo. Yeah, this would allow you to test these directly. So what we have done is we've looked at a bunch of lung epithelial cells on the market that's available to see what their how like they are to the lung in terms of bioactivation, in terms of toxicity related parameters, and then they vary hugely. So there's going to be some challenges, and most people for the lung they would use like B2B cells or something, the primary cultures. It's still unclear how well those represent the real lung, because I mean you can't really detect P450s in those cells very easily. So but this direction, I think it's something that's really worth pursuing, and I think with the new technologies in 3D culture, I see this as a potential something that will help us. Yeah. So thank you.
Thank you. Thank you.
Yeah.
I think Jiahui has a question.
Yes. Hi, Dr. Ding. Thank you for your excellent presentation. So I'm an assistant professor and I'm working on how the air pollution affecting the fetal lung development. So in my experiment, when I exposed the pregnant mouse to benzene during pregnancy, and I noticed that the very critical enzyme called CYP2E1 get really greatest inhibition in the fetal lung. So I'm trying to think, is this a protective mechanism for the fetal lung to, trying to not generate the toxicants in the fetal lung, which leads to a very detrimental impact? What do you think about the CYP450 enzyme as a,
Yeah, that's a very interesting question. I studied CYP2E1 for my PhD thesis. So it's it's long known that alcohol would induce CYP2E1, but also inhibit CYP2E1. So I think you have benzene would be the same way, because CYP2E1 is induced by its substrates, and inhibitors in that sense probably work the same way, because you bind to the protein, you stabilize the P450 against protease degradation. So is it protective or not? I think that's a tough thing to say, because you may inhibit the enzyme activity in vitro, but the remaining activity, you probably will have an increase in protein levels because of the induction aspects. And then over time, over a sort of a daily, longer exposure, you may actually have more capacity to metabolize, to bioactivate benzene. And 2E1 is actually the enzyme, most important enzyme probably, or if not but one of the most important enzymes to bioactivate benzene to the carcinogenic forms. So I wouldn't say it's protective. Certainly, I wouldn't say we can use benzene to protect. But I would look at, because inhibition, unless if it's a mechanism-based inhibition where you actually kill the enzyme, and that's a different story, right? But benzene, I think by large, most of the inhibition may be because it's a substrate. It's a competitive inhibition. So it's a transient inhibition. It's not permanent. I would look at induction, and I would look at overall production of benzene metabolites, like conjugates or something, as a biomarker of bioactivation, to see if you had the increase in bioactivation overall or not.
I see. So the critical is also to see the metabolite actually accumulate in the fetal lung to explain the enzyme activity.
Yeah.
I see. Thank you.
Other questions? All right, I'll try to stumble through something here. So we've got some chemicals that it looks like the metabolism in the lung is probably most important, and we've got some others where maybe metabolism in the liver, producing metabolite that travels to the lung, is very important. Seems like this is a complicated issue, and as you're getting into, I mean, do you think part of this could be predicted based on the enzyme profiles and their abilities to metabolize particular substances in the two tissues? And then on the other hand, I would think maybe it's also in part a function of just the particular metabolite and its stability. Maybe there are some that can make their way through the blood associated with albumin, and there are others that are just so reactive that they would absolutely have to be produced in the target tissue. Can you just give just some thoughts on those possibilities or any others you might think
Yeah,
of?
these are excellent points. I think some of the modeling approach would definitely help. This chemist, I'm not a chemist, and I can't, I haven't done this myself, but chemists have ways to calculate the reactivity of a compound, like how energy, what's the level of energy of the compound. And one guy was showing me that using that as a way to predict whether something is going to react with DNA or protein. Very little is known about what determines the ability to transport through the circulation here. And in fact, it was surprising to us that the albumin had anything to do with this, because when the albumin actually forms covalent adduct with naphthalene oxide and to less extent with the quinones and so on. So we actually was detecting that adduct and not thinking about the covalent binding. But I mean, it's certainly it can bind to it. Lots of things bind to this. So there's a lot to be learned about this. I mean, not to predict this ability to sort of cause trans-organ damage, then we really need to know what controls this transit from one organ to another organ. So yeah, to answer your question, I think the chemical structures definitely is important in there. And once we know which metabolite can transport, and then I think knowing which which enzymes are involved in the formation or degradation of the metabolite, then we can sort of do modeling to predict the risks of that. So it's complicated. I know. That's why I feel like that's why people haven't touched it that much, because it's not easy to track.
Right. Yeah. Thanks. Other questions? I'll just ask one maybe trivial question. I'm just a little intrigued. So you made an actual albumin knockout mouse, or is this human?
We actually bought this from Jackson Lab.
So I was just curious what kind of shape they're in, because I would think a mouse would.
That's a great, yes. In general, they're fine. So there is some changes because albumin is involved in I think what's the IgG homeostasis, like the binding to them. So there's some changes in that, but it's by and large they're normal. There's not huge. I mean, there are people actually. There's a genetic polymorphism. There are people who don't have albumin.
Wow, I didn't even know.
So, and they seem to be fine. Under normal circumstances. I don't know what happens when those people get put on a drug, right? I mean, because the whole pharmacokinetics would be different without albumin. So,
okay, thanks. Other questions? If not, why don't we give thanks Xinxin one more time for a very interesting talk, and we can conclude this seminar. So thank you.
thanks everyone.
Nice seeing you, Xinxin. Take care.
Yeah. Bye.
Bye.